For our next and final lightning talk, I have the pleasure of introducing Heather Lynch, here from Stony Brook. She's an associate professor in the Department of Ecology and Evolution and is jointly appointed in the Institute for Advanced Computational Sciences. Her background is actually in quantitative ecology, and she has a Microsoft and National Geographic Award for her work on studying Antarctic wildlife populations.
Well, thank you so much. This is a great opportunity for me to share a little bit about what I do, which, as you'll see, in many ways is very different from what all of you do, but I think in key ways shares a lot of similarities. So I like to motivate my own work in AI by just highlighting that much of wildlife biology today would look pretty familiar to Darwin. And to borrow a phrase from a certain someone, I think that's pretty sad. Because when we think about the kinds of advances we have now in all areas of science, from precision medicine to CRISPR to genomics, wildlife biology just really hasn't kept pace. And that's too bad, because environmental sciences as a whole has evolved over the last century. And for example, if I asked you, what the temperature was down in Antarctica, you sort of know implicitly that you don't have to run down there with a thermometer, that there are these automated weather stations all over Antarctica that are streaming that data back to the internet, and you can pull that up on your desktop. And likewise, with satellites, we have the ability to map things like sea surface temperature over the entire globe and to get that information back in your office. Even things like wave height and wind speed, or ocean color, which is a measure of biological productivity in the oceans, all of that can be mapped at very high spatial resolution over the entire globe and delivered to you on your desktop. But if I asked you where all the penguins were, or any other animal for that matter, we have no comparable data sets on wildlife at all. We simply just don't know where the animals are. We don't know how many animals there are. And in fact, we don't even have any real tools to get at that question at the scale where we have information from other systems. So my work is, as was noted in the introduction, in Antarctic penguins and Antarctic wildlife. And you can see this is just one part of an island in the middle of the South Atlantic, one of the most remote islands in the entire world. That as a penguin biologist, you've got a number of challenges, which is that actually just finding your animals is really difficult. Once you've found them, getting there is very difficult. And even once you're there, counting them is very difficult. And the traditional means of counting animals, literally one by one, just the way Darwin would have, simply doesn't work for populations this big. So my research has been very focused on alternative methods of surveying Antarctic wildlife, starting with our work to survey Antarctic penguins. And the way we do that is using satellite imagery of the Antarctic. And in satellites, we can't quite see individual penguins, although it's awfully close. But what we can see is that pinkish red guano stain that's left by the colony. And guano being the technical term for penguin poop, essentially. You can see that the penguin guano doesn't actually get very far from the penguins themselves as they're incubating their nests. And so the guano stain, which is visible from space, is the exact size and shape of the colony itself. And so we can work backwards to tell how many penguins must have been nesting inside that colony. But manual hand annotation of imagery simply doesn't scale. I mean, this is a lot of what I've been doing over the last decade, is looking at high-resolution satellite imagery and literally just drawing polygons around guano stains. But there's just simply no way that I can continue to annotate the entire Antarctic coastline on anything close to a regular basis. And so my lab has made a huge investment in AI and machine learning to try and train computers to do some of this work. And the challenge is, as I'll show you in a second, this very high resolution imagery that we have does not, the traditional methods that remote sensors would use do not work. And so the imagery is in fact so high resolution that it starts to look more like a computer vision problem. So just to show you what guano actually looks like on a high resolution satellite imagery, you can see the guano stain there along the coast of this island. It's that kind of pinkish red guano stain. This is sub-meter commercial satellite imagery. So in this image, every pixel is about the size of a piece of paper. It's a 31 centimeter resolution image. But the challenge is that if I zoom into an individual pixel, the penguin guano actually doesn't look that different from some of the bare rock areas. So at a pixel level, there's not enough information to make that distinction, and the way that I do as an expert annotator is that I can take the context into account, because I know that penguin colonies are along the coast, and I can incorporate things like texture. So we need to think about alternative methods like deep learning and convolutional neural networks, as I'll talk about in a second. So I think of this high-resolution imagery as being like the sirens. You can think back to your humanities classes. You know, the sirens are calling to Odysseus as he's in his ship sailing past, and they say to him, you know, come this way and stay your ship so that you can listen here to our singing, for no one else has ever sailed past this place in his black ship until he has listened to the honey-sweet voice that issues from our lips. Then goes on, well pleased, knowing more than he ever did. Over all the generous earth, we know everything that happens. And so I love this quote from the Odyssey because, you know, this high-resolution satellite imagery for me is the sirens. Like, it promises universal knowledge about what happens all over the planet. And yet the question is, as a penguin biologist, you know, obviously my career has steered towards these rocky shoals as I try and, you know, integrate some of these AI techniques. And I just hope that, you know, the ship does not crash. The reason why I actually worry a little bit about the ship crashing is that finding or teaching computers to find guano in satellite imagery is a very difficult problem, much harder than we had thought when we started it. A couple of the reasons are that we have very small training sets. We talked a little bit about that earlier this morning. There are very few experienced annotators, like maybe 2 or 3 in the world, and there's only so much time folks like myself can spend providing high-quality annotated data sets on this high-resolution imagery. We also, as I showed you, have a lot of false positives. So there are a lot of things that look like guano that aren't guano. And finally, we have, you know, a variable background substrate. So what I mean by that is that even if we learn what the colony looks like, the penguins are nesting on rocks that themselves change in character, both through the seas and in between sites. And so that makes it difficult to train models that are generalizable. How we make this easier are 2 tricks that I think have a lot of application outside of this work, as you'll see. And one is that we can use the site fidelity of the penguins. Penguins are extremely site faithful, and they go back to exactly the same nesting spots year after year. So as a human analyst, one of the things that I'm using is that I'm looking for the colony in the same place where the colony was last year, because that's actually the most salient piece of information I have about what's actually guano and what's actually rock. I can also use the classification of lower resolution imagery to train a model for high resolution imagery. And in fact, this was introduced in the keynote talk, this idea of a weak annotation. So if you think about, you know, a time series of satellite images that we might have for one of these colonies, and those red blobs are the guano stain that I'm trying to track through time, you can see that the guano stain might grow or shrink over the course of time, but essentially it's in the same location. And if I can find a way to harness the power of that connection through time, my models are going to work significantly better. And we can do that through what are called recursive neural networks, which some of you may be familiar with. And as a Bayesian analyst, I kind of like this very Bayesian idea that we have some prior expectation for where the guano is, and we should find a way to do that. And these RNNs provide a mechanism for doing that. The challenge is that if I'm looking at this time series of images, I need some way of aligning them very, very carefully so that a pixel in one image actually maps onto the correct pixel in the next image. And it turns out that that's a very difficult problem in and of itself, because while the guano stain is changing, genuinely changing in size and shape over the years, we have these artifacts due to what's called orthorectification, which is what happens when you take a 2-dimensional satellite image and you're trying to drape it over the landscapes, over the hills and over the valleys. And it introduces these artifacts, which means that the morphology of that guano stain is changing, but in ways that complicate your search for real change. So one of the challenges, one of the ways that we've been trying to address this challenge is through what are called shape encoders. And I won't get into the details here, but you know, I think of it a little bit, you know, as a principal components analysis. You know, that we can encapsulate the shape of the guano in the prior year and put that into our models so that our models basically know what shape object it's looking for. And then we can combine that with this really high resolution information that we're getting from the current year, which provides all the color and the texture that we might use to classify penguin guano. So combining these shape encoders with these recursive, you know, in a recursive neural network provides a lot of additional power for our models. The second is this weak annotation trick that was mentioned briefly earlier. And the idea is that, okay, let's say that we have another data set with lower resolution imagery. And in our case, we do, because the Landsat satellite image, which is several orders of magnitude coarser in resolution, we have figured out ways of annotating that quickly. And so if the yellow here is the guano stain in the Landsat imagery below, and I know that about 4% of that green patch is guano, then I can train my model to look for something that is also 4% cover in the high resolution image. And over thousands of these weak annotation pairs, we can train the model for high resolution imagery. So here I'm just putting this up here to show how complicated it can get when we try and pull together all of these different threads, these recursive neural networks and this weak annotation, all of this just to try and count how many penguins there are in the world. You can see this is a diagram of the rocky shoals on which I'm attempting to crash my career, but it turns out to be an extremely interesting computer vision problem. We can do this not just for penguins, but also for other animals. We've developed models to find pack ice seals in imagery, also whales just underneath the water. In fact, it turns out that anything larger than a piece of paper we can now find in high-resolution satellite imagery, which is most of the charismatic megafauna on the planet. So just to close, I just want to highlight how far we really have come. And several years ago, National Geographic came to me and they said, Heather, we've got a question. We're writing this big feature story on the western Antarctic Peninsula. Can you tell us how many penguins there are in this region and how their populations have changed over time? And first of all, as a wildlife biologist, I was like, oh my God, I thought you'd never call. I was so excited. But then I thought about it, and if they'd asked me 10 years ago, that would have been an entire PhD dissertation. But with the advances that we've made with AI and modeling and decision support software, I was able to get them the data that ultimately ended up in this figure in just a matter of minutes. And so on this continuum of a dissertation to a couple of minutes, we've really made great strides in our automation of surveying the planet for all of its animals, and eventually we'll reach the scale where the animals and the environment can be monitored together. So with that, I'll just thank my funders, and I look forward to the panel. So thank you very much.